Good afternoon, everybody. This is a seminar in the Biomedical Informatics series. I don't know, is this the last seminar in the series? For this academic year, this is the last seminar. And my name is Alekseyenko. I'm a junior faculty at the Center for Health Informatics and Bioinformatics. And I'm leading the Microbiomics Informatics Lab since 2011. And I'm also an Associate Operations Director for our consulting service. So today I'm going to talk about microbiomics, and I usually like to start this by showing this picture taken in Vancouver area because it demonstrates so many different habitats that this Earth has, and every habitat harbors a unique set of communities of microbes, and these usually are distributed according to gradients, and there are many of such gradients you can imagine, as in this example, whether there's more or less light, or here whether it's by proximity to water, or here whether it's more moist or more dry, the variations of moisture, and here along the body of this sleeping fisherman who happens to be my friend. Of course, in the medical center, we are more interested in how these communities are affecting our health, and a big push to this effort was given by the HMP Project by the NIH, which set out to determine whether individuals have such a thing as a common human microbiome. And it came, some of the goals of it was to understand what changes in the human microbiome can cause, or at least be correlated with changes in human health. And then, of course, a lot of the effort was given to the bioinformatics of the microbiome, because it's actually the technology that has enabled us to study these communities at a greater depth. And associated with technology, you need additional methodologies to be able to get a sense of the data from these high-throughput methods. And then another goal was to address ethical, legal, and social implications. I'm not going to talk about these. So if you think about these microbes, this is just sort of a reminder of biology 101. So what's happening there? And the mechanisms that underlie these microbial communities are governed by interactions. And you can think of these interactions as the typical ecological interactions: symbiosis, competition, parasitisms, and with additional complexity that you have a host, an environment that's not constant, that's changing, that also interacts with the microbes and competes for nutrients that we intake. And we have to, I mean, it's a fact that whatever we eat, we don't decide. It's the microbes that are in our gut decide what we actually get to eat from our food. Then absorption and processing of the hormones that are in the gut, happening in the gut, is also governed by the microbes, and we interact with them in this sense. And of course, this is a big one. It's the immune system that constantly tries to fight out all the invaders, and there exists a balance. Exists an imbalance between the communities and us. So what are we trying to do? So in essence, we're trying to look at phenotypes, and then we're trying to look at microbes and see, does a particular phenotype, a continuous phenotype, for example, such as weight, is correlated with presence or absence of particular types of bacteria, or maybe sets of bacteria. Maybe it's not just one particular bug that is associated with the phenotype you're looking at, but a more complex structure that you can identify. And more globally, you're looking at associations of phenotypes, then different host factors, such as genetics, inflammatory, metabolic, and immune system, and the microbiome. And typically in the experiments, one would know and can directly establish this association between the phenotype and the host variables. And then you would hope to get the association with the microbiome. And the most exciting associations that you want to establish is where these interactions between the phenotype and the host genetics, inflammation, or metabolic states is actually mediated by the microbiome. So this is a sort of a causal question that people try to address in these studies. So NYU is a very nice place to study this, and in particular when I came here in 2009, NYU has already been a site for 2 HMP demonstration projects. These were the projects directly funded by NIH, and they worked in collaboration with NIH to give initial evidence for these associations. And the 2 projects were the evaluation of cutaneous microbiome in psoriasis with the PI, Dr. Martin Blazer, and the other project is the foregut microbiome in the development of esophageal adenocarcinoma with Dr. Jihen Pei. Then when I came here, we have also at the same time established our consulting service, and I was very active in this service, and sort of I saw the trend that every year we would see 10 to 20 new projects, new proposals that would want to study this or that aspect of microbiota. And of course, none of them were successful, but a lot were. And we're sort of slowly emerging as a very strong site for the microbiome research. Hopefully nationally, taking with the Human Microbiome Initiative here that has been announced this year, we're becoming a very important site for microbiome research. So what is it that has allowed us to study the microbiomes? I've already mentioned that it's technology that is important, and in particular sequencing technology, because it is through sequencing that we're learning what these microbes are, what their genetic composition is, and in particular, the 2 systems that are important for this study is the 454, which was at the forefront of the high-throughput microbiomics. People used amplicon sequencing to get a really depth census of the microbes, and then lately it has become the MiSeq, slowly replacing the 454 technology. And then the Illumina HiSeq or GA2, these were also instrumental in getting first information about the genes in the whole gene sequencing efforts from these communities. So here's the idea how we can learn from what organisms compose a particular microbiota. So we do this by using a targeted sequencing of a marker gene. And a particularly useful marker gene is 16S rRNA gene. This is a gene that is ubiquitously present in all bacteria, and it has these regions of highly variable regions shown in yellow, and then it has constant regions shown in blue. So the importance of this is the following. The constant regions allow us to target specifically particular areas of the gene because the technology does not allow to sequence the entire thing. And the yellow regions have enough variability such that genes from different bacteria can be separated from each other. So so the Human Microbiome Project evaluated using different regions of this gene for identifying microbial communities and and most of the census studies were used using this particular marker gene. So what happens informatically is that you collect your samples, you perform sequencing, whether by 454 or MiSeq, and what you get in the end is a set of sequences that are tagged by samples. So these yellow things represent a tag, so that you can sort the sequences into the samples, the columns of this data matrix that you're trying to reconstruct. And then from these sequences, you try to identify which microbes they are and the abundances of each microbe in each sample. So the upstream informatics in this problem deals with reconstructing this matrix, which you can use for further analyses. Currently, there are two main directions in how this happens. So it's so the most dominant approach is the reference-based OTU construction, which constructs OTU based on their similarity, sequence similarity to each other. You set some thresholds of how similar sequences you would treat as a single organism, and then when you see these clusters of sequences with a given similarity, you call them an OTU, an operational taxonomic unit. And then there are a number of different approaches to doing this. I would like to highlight this method, SIMRank, which was developed here in part at NYU by Dr. Jahan Pang, and in collaboration with Todd DeSantis, who's now at Second Genome, a company that does the microbiome studies commercially. And then another approach for constructing this sort of feature table is based on phylogenies. So instead of using similarity cutoffs, you actually reconstruct the phylogeny of each individual sequence and then use monophily in the tree, so these monophyletic groups as the definitions for your OTUs. This approach is very promising. It's more precise than this, but it's rarely used because of the computational complexity of it. However, even with the reference-based OTUs, you do typically reconstruct phylogenies in order to see, to judge how similar different taxonomical units are to each other, and whether you may merge, or whether two OTUs that you're looking at are might actually be the same thing. Another mode of studies in the microbiome is to use whole genome sequencing to get a sense of what genes are present in this meta community. The 16S only allows you to hopefully answer the question, who's there? And the whole genome sequencing approach aims at trying to figure out what they do, so what functions are present there. The informatic approach is the upstream informatics is quite different from with the whole genome sequencing. You start with short DNA sequence reads. You do some basic pre-processing. You align them. You try to assign functions to by aligning it to known databases, and then you try to assign weights to these functions sort of corresponding to abundances. So how much of a particular function do you see in a particular sample? And this diagram shows one particular method of how to do the upstream pre-processing. This slide was prepared by a graduate student who works in the Microbiomics Informatics Lab and is shared between Blazer Lab. But in the end, you end up with the same data matrix as you do with the census data, with the 16S data. And the methodologies for analysis of these matrices are quite similar. So I will focus mainly on analysis of the of the 16S data, but you can keep in mind that the very similar methods can be applied to 16S data, and they are applied. And in fact, Francis Kerrigan is preparing a very important study in how to apply these methods and how to learn, actually extract useful information from them. So. So let me tell a little bit about the Microbiomics Informatics Lab. So it's part of the Center for Health Informatics and Bioinformatics. The lab was organized in 2011 as part of sort of developmental plan for CHIBI, and that year was particularly rich for new labs. There was a proteomics lab developed established that year too. So we work closely with other labs in the center. In particular molecular signatures and computational causal discovery labs, and obviously we work very closely with sequencing informatics group headed by Stuart Brown. We collaborate a lot with investigators across NYU. Most of the projects that we do in the Microbiomics Informatics Lab are initiated by individual biomedical investigators in different departments. We provide education to NYU community. We have taught a course last semester on a tutorial course in Microbiomics Informatics, and we're going to teach it again hopefully next year. And then there's also a course that I'm teaching for a third time in a row, which is outside NYU in Seattle in the Summer Institutes for Modeling of Infectious Diseases and Summer Institutes for Statistical Genetics. So that also gives some publicity to the microbiomic efforts that we have here at NYU. And obviously we provide mentorship to graduate students at NYU. Francis Kerrigan is one of the first microbiomics students in the informatics lab, and then I'm also sitting on committees of multiple graduate students. And training to junior faculty, we have a number of people either applying for K awards or having K awards that have microbiome studies as part of their course, and we work with them to help them with their studies. So that said, let me mention some of the methods that are very common in analysis of these data. And this is sort of, this is based on the many studies that I have personally performed and the literature. So the number one method for these analyses are methods based on multivariate decompositions. And in the next slide, I'm going to give a brief overview of what that means. There's obviously the classical bioinformatics techniques such as hierarchical clustering or discrete clustering. Discrete clustering has become very popular in microbiota after a paper published describing enterotypes in the human gut, where you have different, seemingly different microbial types in different individuals. And from that spurred a number of other types of microbiota, cutaneous types, pneumotypes, microbiota in the lung, and so forth. Obviously, univariate analysis, it's a very rich area because people don't really want to know that you have an association of microbiota with a disease or some metabolic state. They also want to know, is there a particular bug that will give me that information? And the univariate analysis is what gives you that ability. The area is also problematic because oftentimes you don't know any parametric form for the distributions of the data. The data that you get is typically compositional, so you have correlations and your tests will be correlated. So that motivates the importance of multiple comparisons and specifically tailored for these types of data sets. Then my favorite set of techniques is this ANOVA and MANOVA multivariate analysis of variants, and especially non-parametric. These ones are the ones that allow you to give sort of the ballpark answer to these questions. Is this factor associated with the microbiota? Is this factor conditional on some other factor associated with the microbiome? Are there components, how you can decompose the microbiota between the multiple measured clinical factors? And then something that I will mention today at the very end is some efforts at predictive analysis, actually trying to build classifiers to develop biomarkers and to measure productivity of these biomarkers based on the 16S community data. So, as promised, here's a brief description of the multivariate data analysis. So the idea behind this set of methodologies is the ability to compute distances between arbitrary objects. And these objects that we are dealing with here are the microbial communities. So I can compute distances, how different, you can think of this, how different the communities in the gut are from the communities in the hen, or how different the communities in my body are different from the communities in someone else's body. So then, using these distances, we're able to reduce dimensions to make this data more simple by embedding it into Euclidean space. And then utilize the more general framework, which is called duality diagram, which uses the data and then and the sample and feature weights in order to collapse the data into this duality. It was called duality diagram, where you can look at associations, multivariate associations between your features and your sample factors. Principal coordinate analysis is one that's been popular in the microbiomics area because it can utilize partially the duality diagram through what is known as unifrac distance, which collapses the OTUs using the phylogeny information. So in essence, it incorporates the phylogenetic information in its analysis. Of course, I said that it only partially uses the power of this methodology, and the reason is because the unifrac produces distances between samples, and it completely removes any information about the features, the OTUs, so that you can only work in sort of part of this duality diagram only in the sample space without being able to associate it directly with the feature space. So there are some efforts in the Microbiomics Informatics Lab to overcome this difficulty. I'm not going to talk about these, but the idea is to utilize the same information about the phylogeny, but not through the distance metric itself, but through the column weights, and we are able. Our first analysis showed that we're actually able to do that, and we're able to produce analysis at least as good as the unifrac. So this is very promising thing. I'm very excited about this. So now I just wanted to go over a few successful projects that the lab has participated in, and one of the biggest projects that we took part in, and one of the first by time, is the study with the Blazer lab on antibiotics in early life, where we had a mouse model of constant exposure, subtherapeutic exposure to antibiotics, similar to what is being practiced by farmers today. And that's why you see oftentimes in the stores no antibiotics used on a lot of the meats, is because they're concerned that these antibiotics may be doing something that you don't want to. And in fact, they do. So what we looked there at is mice that were on constant subtherapeutic antibiotic treatment throughout their life. They were actually started on antibiotics before they were born with the mothers. And what you see is that although the differences in weight, in total weight, are very minor, if you dig deeper, you see that there's a lot more fat mass in these mice, and percent body fat is increased. And these are the MRIs of these mice, and you can see this is the control mouse, a lot less fat deposits than the mouse that was treated by subtherapeutic antibiotics. Also, you see differences in developmental phenotypes, such as bone mineral content at 3 weeks. You see a lot more density in the bones of these mice. So what we did, we sequenced the gut communities of these mice, and we tried to see whether there are specific microbes that can be associated with subtherapeutic antibiotic treatments. So basically, what does this stat hit in the gut? And in fact, using the univariate testing methods, we were able to find a few differences in the fecal samples. This is these first two columns, and a little less in the cecal samples. We also see these differences when we in particular we look at the ratios of different taxa, such as bacteroidetes and firmicutes. We see decreased ratio in the stats than in the controls of bacteroidetes to firmicutes. So effectively, I also have to mention that in additional experiments, we have seen that there's a modification in fatty acid processing that is present in the stat samples compared to the controls. So in essence, what we have shown in this study is that subtherapeutic antibiotic treatment affects developmental phenotypes, affects metabolism of fatty acids, increases acetate butyrate and propiate production in the samples, and also has a difference in the microbiome. An interesting question that is for further analysis, is for further studies, is whether the microbiome is somehow related to these processes. And our guess is that it is, but we have not shown this directly in this study, in this experiment. Our other study of antibiotic treatment models included a post-antibiotic treatment model as compared to STAT, which is a constant antibiotic present in the diet. This one mimics what infants get, for example, for treatment of ear infections. You get these pulses of antibiotic, and then no antibiotic, and then you get another pulse. And this study demonstrated how we can effectively use the duality diagram and in order to study the dynamics of these communities. Unfortunately, this does not reproduce really well on the screen, but if you can follow me, what happens here is that when we start the experiment, we give the first pulse to the mice, and all the mice were started from this cluster, so from this area. And once we give these mice the antibiotic, the mice that receive tylosin have all moved to a different area representing a different area in the sort of different microbial community state. But the mice that received a different antibiotic, amoxicillin, stayed with the controls. With the second pulse, the tylosin group has come even further, and some of the amoxicillins have also moved to this sort of antibiotic response area, as well as with the third pulse. And then the second part of the experiment was to change the diet of these mice, put them on high-fat diet, and then see what happens there. And then once you put it on the high-fat diet, you see that the control mice have suddenly moved to this red area. This is the area in the microbiome space that represents the high-fat microbiota state. And while the antibiotic mice still remained in their own area for quite a bit of time, and only in the very end they all converged into one area. So note that the mice in the antibiotic groups have stopped receiving antibiotic at this time. So they were trying to see how the microbiota recovers to the same area. So as you can see, this is a very graphic, very powerful technique for looking at what happens with the microbiota at large due to these interventions. Another interesting project that was just recently published this year is where we studied the cutaneous microbiota in diabetic and non-diabetic men. The problem here in this study that motivated this study was that a lot of diabetics unfortunately end up with amputations because of ulcer development, especially in their feet, and specifically in their feet. And so we wanted to study whether there's early signs of potential changes in the microbiota between the diabetic and non-diabetic men. So we enrolled subjects that did not have any ulcers yet, and we studied their microbiota on their hands and on their feet. And we compared them to a group of controls that also had samples from their feet and hands taken. So here's another example of using the duality diagram. This is PCOA plot based on weighted unifrac distances. So when the first graphic shows the microbiota in the diabetic and non-diabetic men on their hands, and you can see that so each point represents a sample, and circle shows sort of a center of mass for a particular group, and the maroon is the diabetic, and green is the control. And you can see that the general area where these samples reside overlap completely. So you can see no difference in the microbiota in their hands. However, when you start looking at their feet, you see a separation starting to develop on the first principal axis here. So the microbiota of the diabetics has moved to the right, and the microbiota of the diabetics moved of the controls moved to the left. So even though they don't have any signs of ulcers yet, you already see differences in the microbiota, and this is a reflection of changes of their metabolism, and especially their ability to control temperature. That first shows up in the extremities, such as the feet. So this is one thing to note here is that is these numbers that are written there in very small print, is the amount of variability in the microbial data that the axis can explain. And it goes up to 54% on the first first axis. So that says that this is, the difference between diabetics and controls is the major factor that's there, that's present there. And this is very rare to see in other studies, especially studies in of the skin. There you see the numbers being very small, meaning that there are many more factors that are contributing to the variability of the microbiota. And you will see this in this study, other study that's currently trying to publish, and this is a result of the demonstration project on the cutaneous microbiota in psoriasis. So here, excuse me, here the experimental design was as follows. We had psoriasis patients with visible lesions, diagnosed lesions, diagnosed by their severity, etc. And we collected skin swabs from these samples from the area of the lesion and then from the contralateral site on their body, so that we have a sort of an internal control for these. And we also had a control group which were sampled at standardized sites. And the controls were matched by age, gender, and site, and ethnicity to the subjects. So in the end, we were analyzing triplets that we marked as lesion for the sites that are from the psoriasis site, normal from the contralateral site, and the controls. So when you look at the PCOA there, it's a mess. You cannot figure out anything. So, so based on just the placement of the dots, I never bothered to put the circles in because they would only confuse you. So you note that this number it starts to get very small, 14.5%, and this is not the smallest you can see, depending on how exactly you perform this analysis. So there, first of all, you don't explain a lot of variation using this method. So there's a lot more going on there. Then you see these. So these are the percent variation plot. If you take more and more axes, you don't actually explain a lot more. So the variability is spread out everywhere. So although we do see on the principal coordinate 1, we do see difference between lesion and normal and lesion and controls. It's not a very good method in this particular case. It doesn't really show you that there is something going on, some difference in the microbiota, or especially what that difference might be. So here we applied a different technique. We did discrete clustering of these samples. First, we tried to establish how many clusters they are, and based on our criterion, we found that the maximum variability partitioning occurs with 2 clusters. So we were lucky here. You only have 2 clusters, 2 major states that can explain what's going on. So we clustered the data into 2 clusters, and what we see is that cluster 1 is dominated by proteobacteria, and cluster 2 is dominated by firmicutes and actinobacteria. There's also evidence for differential prevalence of these cutaneous types. We call these clusters cutaneous types. Between the control, normal, and lesion, there's a preference for cutaneous type 1 in the controls, and preference for cutaneous type 2 in the lesions, and they're evenly distributed between the normal in the normals. And also you see evidence for this bimodality in the distribution of proteobacteria, actinobacteria, and firmicutes, the major phyla, which is additional evidence that this is a real effect that we're trying to capture. So this was sort of an exciting outcome. So but that only shows that, okay, there is a way to associate the cutaneous microbiota with psoriasis status or types of skin. So next we try to look at whether particular skin genera will give us any predictive signal for distinguishing the controls versus normal, controls versus lesion, or lesion versus normal. And so we did find a few phyla, genera, that were statistically significantly different between the three types of skin. And then we took, the four major ones, the four major skin genera, surprisingly, were not significant. And these are Corynebacteria, Propionibacteria, Staphylococcus, Streptococcus. When you talk skin, these are the four genera that people are mainly looking for at sort of as markers of what's going on with the skin. They were not significant. So when we looked at the classification signal as measured by AUC, Streptococcus, Staphylococcus, Corynebacteria, and Propionibacteria, they gave signal very close to what you expect by at random. This was very surprising. So we started to look for maybe there is something more. And the way we finally figured out what's going on is that these four genera actually act in concert. And if you add up all the four genera, the abundances of four genera, you actually get pretty good classification with AUC about 67% for control versus lesion. So that was pretty good. And also we see there was a longitudinal component to this study where we had a smaller set of samples that were measured at 3 different time points. And when we looked at in this set of samples, there was no stability even in the control samples if we looked at these genera individually. However, when you combined we combined the four genera together and looked at this additive abundance of the four, we see that it's very, very constant over the course of the experiment. And there is some variability in the normal and lesion samples, which we associate with the treatment, because the psoriasis subject in this experiment were under antibiotic treatment. So the bottom line is that it's not very simple here. It's that you have to really look very carefully, and the simple univariate classifications will not necessarily give you an interesting result or actually lead to anywhere. So we tried to capitalize on the strength of the Molecular Signatures Lab and Computational Causal Discovery Lab and further this analysis and used more advanced classification techniques in order to study these samples. So we, in this paper that's also submitted also on a very similar data set, we also had data from a different 16S locus. So you can imagine that you can produce census of the 16S based on different primers, and they may produce different signals. So we studied that aspect as well. So what happens there is that with a modest number of features, using feature selection, maybe so 3 or 2 features, you're able to produce classifications that are very strong at 0.9 is the 0.89 is the top classification signal. So there's a big promise for using more advanced classification techniques in order to study this. Of course, in order to be able to apply these, there are some restrictions that had to be met. In particular, in sample size, we could not use many of these techniques in our mouse studies because the sample size was pretty small. Although we did have many samples in total measured at different points in time and different conditions. What matters is the sample size measured at a particular point in time with the same conditions. So per class, sample size was pretty small. In this case, 50 subjects per group seem to do the job, and you're able to pick up signals that are quite significant. So another conclusion here is that it does matter which locus you look at, because at the V1, V3 locus, the signals are quite small, and there's in fact only one classification task that achieves a significant result with more features. So it's important to use the right primers for this analysis. So this additional plot demonstrates the different feature selection techniques and the classification signal they achieve based on the number of features selected. And the bottom line for this plot is that you can achieve very compact signatures using the generalized local learning techniques, which are methods based on causal learning, and they have some causal implication for the features that they select. And so in a battery of different feature selection techniques, the JLL techniques produce the most compact sets with the highest predictivity. So speaking of which, we're also trying to generalize even more and look beyond just one data set. We're trying to develop guidelines for how exactly to perform these classifications, what feature selection techniques work better, not just for one specific data set, but in general. If you take a survey of different data sets from different labs, different classification tasks, what happens there? So in this paper we had, and this was just published in the Microbiome journal. It's a new journal that publishes papers in the field of microbiomics. So we were pretty happy about this, one of the first papers to be published in the journal, and they had a pretty thorough selection because they want to establish themselves as a very good journal. So in this study we had 8 different data sets and the two different classification tasks. One was to classify the different body sites. So you look at the microbial communities from different body sites and try to guess which body site it's coming from. So it's trying to build predictor for that. And then the other classification task, which is more relevant to the medical research, is to try to classify disease status based on these data sets. And what these plots show is the average performance of the different of a vast array of classification techniques for these, based on two metrics of how good the classifier is. So you can see that a lot of classifiers have very similar performance if you do not perform feature selection, and in fact these sort of classifiers have pretty much the same. Pretty much all of them have the same, statistically the same performance. Then if you do feature selection, the picture changes slightly with less features. First thing you notice is that you get slight bumps in classification performance in some of the for some of the methods, which happens for both of the measures of classified performance. And also some of the classifiers are really starting to stick out as statistically different from the rest. So we intend to continue this study and expand it more and learn how exactly to work with this data modality. So I think by this time I've bored you with talking about what we did. So I just wanted to summarize briefly where we're going next. So we are going very strongly in the area of predictivity, being able to build biomarkers, classify data sets, produce something that we can use for further prediction, use for development of treatments, development of diagnosis, etc. We're looking also at integration of new data types. So these, most of these results were based only on the 16S data, but there are other high-throughput modalities that will allow you to learn more about the interactions between the microbes and their host. You can study enlarge the metabolites that are being produced, the gene expression in the host, or host metabolic responses specifically, immune and other assays. To use these in conjunction with the 16S sensors data would be very useful in order to get a better picture of what's going on. Then another area where we're trying to make progress is to incorporate in these types of analysis the existing knowledge about pathways and disease area knowledge in a seamless way. And very small area that sort of sings to my soul because of my background in phylogenetics. And I've mentioned this, I'm trying to work out how to incorporate these phylogenetic relationships in distance-based methods properly, such that you can use the duality diagram in its complete power. So that's another area where we do some work. And of course, there is methodological work that spurs from more advanced experimental design. People are moving more and more towards time series analysis because the sample sequencing becomes very cheap, and it simplifies the control because you sort of have an internal control. But it also makes a little for a more complex informatics analysis when you have time series with these highly multidimensional data sets. And I would like to bring back this point about the causality, where hopefully we will be able to answer these questions that will show whether microbiome, at least in some cases, mediates this relationship between the host and a particularly visible phenotype, host variables and a phenotype. So having said that, I would like to thank my collaborators at CHIBI, Dr. Alaferis, Stadnikov Brown, Michael Hanoff is a programmer who worked with Zhigo Li, Hao Chen, and Kranti Kanganti. Kranti is currently at the University of Texas. Also Francis Kerrigan, my graduate student. We work closely with GTC, Jerry Zavadil. Hopefully the new director will be as collaborative as the old one, and we're looking forward to working with GTC again on many of these projects. And then HPCF, High Performance Computing Facility, Dr. Stathiadis and Peskin, and my collaborators at NYU, Dr. Blazer, Siegel, Ahn, Hayes, Pei, and Dr. Edel. Thank you.
I was struck by that one table where you made that the antigens from B1, B3 versus B3, B5, striking difference. That's a little worrisome for those of us doing these experiments. Is that going to be generally true, or is it different? Or?
I expect it to be different. I do, because so I do teach it in my class. I didn't say it explicitly here in this course. What we're actually doing here is not looking at the microbes. We wish we were looking at the microbes. We're actually looking at the census of the 16S rRNA gene, which is maybe quite different from the actual representation of the microbiota. So, and also because of the peculiarities in how exactly these variable regions vary between different types of microbes and the difference in composition in different areas in the body, you will see the differences between the sites. Hopefully, as you go for longer sequences, as the technology develops, you will capture more of the variable sites in one shot, and then you'll get away from this problem somewhat. Yeah. Yeah.
It seemed that your classification analysis showed improvement when you did feature selection, bringing down just to a relatively small number. Those features are single OTUs?
The features are single OTUs when you select a few of them with the feature selection technique.
Is that correspond to these analyses that use very complex definitions of the enterotype, which is, at the very high phylogenetic level, as opposed to OTUs? So enterotype is like, a high abundance of Firmicutes or something like that, or a complex definition.
Yeah. So enterotype is basically a description of range of how of sort of a particular slice of of all possible distributions of microbes, right? So you have a particular distribution associated with one enterotype.
Right. Your selection process showed the most predictivity when you use just a few organisms rather than one of these big complex definitions.
Yeah, that's true. That's. I mean, you have. I mean, so the enterotype, just by its nature, it captures all. It has to define everything that's present, and of course, there's something that a lot of things that are not important for the phenotype. So if you sort of throw that away and only focus on a few components, you may get a better signal. Yeah.
Is there capability to do whole genome sequencing of a microbiome?
There is, and people do it, but I mean, and it's not, again, it's not whole in the sense that you sequence everything completely. Again, you take a subsample of everything, but you do a good census of the different genes that are present in a particular community. And
the driving at this is can we describe the metabolic output of that bacteria?
Yes, I mean there are efforts to do it. I mean the efforts to sequence the genes, try to figure out which functions are present there, what particular metabolites there might be present, map it into the metabolic pathways. And in our pulsed antibiotic treatment project, Frances Kerrigan, that's her sort of project, is to actually look at that, and she's made a very interesting observations related to kidney stone formation based on these microbiota. So it turns out antibiotics alter the composition of the microbiota and the metagenome such as to favor formation of oxalate deposits. So you can figure out things like that from these data.
Any other classification tests that would be very useful from a medical standpoint? I don't know, predicting survival or and so I guess what are some possible? So assuming we can construct a classifier that achieves really good accuracy, would that be more useful as an automatic diagnostic measure, or more for unraveling the?
Perhaps, I mean there are tests where other data modalities do not produce very strong classification signals. So if you can do it with the microbiomic data, or you can do it in conjunction microbiomic data plus something else, then these classifications will be very, very useful. And if you do it automatically, even more so.
Okay. So that would be as an early diagnostic tool?
Yeah.
Yeah, when we do the longitudinal analysis or time series analysis, is that meaningful to compare the absolute abundance data or actually only meaningful to compare the relative abundance data?
So with these data, it is hard to get at absolute abundances because by the nature of how it's collected, and even if you think deeply, there's a confined space which these microbes occupy, right? There's presence of the compositional effect is present there. So it's very a lot of people are concerned about this problem, whether you should be looking at absolute abundances or relative abundances. However, it is not clear. It's still not clear which way is best to look at.
For the same subject, if the samples collected from at the different time point, is that the absolute abundance is comparable, or actually is a loss of factor?
Probably not. The very simple reason, unless you do some data modifications. So if you do it as raw counts, definitely not comparable, right? But there are techniques that try to infer the absolute abundances from the relative abundances, or sort of work in the absolute abundance space when only relative abundances are present. You can look at this method, SPAR-CC, for example. It's a method by Eric Elm Lab from MIT, I think. They're looking at the data is in relative abundances, but they're trying to make inferences about absolute abundances. The main complication there, when you are actually interested in working with absolute abundances, but you only have relative abundance data, is that your results become correlated. So if a particular taxon is increased in one set of samples compared to another, that means that something else has to be decreased there. So you have this correlations. But there are ways to actually remove these effects, and these ways are based on for example, on renormalization techniques. But the most important thing is that even I don't have proof for this, but I'll give you a conjecture. So if you have a compositional data set and you're interested in inferences about absolute abundances, the components that are significant will not become not significant in the compositional data. So in your final table, if you did multiple tests, right, and you see multiple things that came up as significant, things that have to be significant are still going to be there. So you only have an inflation of false positives. Yeah.
Last question. I'm sorry.
So Alex, I remember when you showed us the duality diagram, you said we just freed ourselves from the feature space and we went into sample space. Mm-hmm. What was the whole point of doing that? Because when we operated in feature space, I mean, you did all the classification and feature selection, and we worked so well in feature space. What was the whole point of going into sample space? When you constructed the duality diagram?
So what I said is that the duality diagram allows you to operate in both feature and sample space at the same time. The results with the duality diagram that I showed, just because they were based on the distance metrics that are not decomposable in feature space, they were only sample space results. Does that clarify it a little bit? Okay, sure. All right. Thank you.